All right, folks, welcome back. How'd you like that compliment in the last section? It's just remember, it's not well, it can be very difficult, obviously, but what we want to do is we want to just realize there's three complement cascades and a common pathway that leads to inflammatory molecules. So if you're overwhelmed by the last section, just remember, look at the mile high view of complement. All right, we're going to keep going now, and now we're going to look at some adaptive immunity or specific immunity. So what we got done doing is we looked at everything you're born with, and we call that innate or non-specific immunity. Now what we're going to do is look at this adaptive or specific immunity. And the difference is that specific immunity reacts to a particular pathogen. It's not like a generic phagocyte that engulfs anything. This will react to Streptococcus pyogenes or Staphylococcus aureus, and it will react a certain way. All right, here are the four properties of specific immunity. So first of all, it's specificity. So that's probably the biggest thing. And the second thing is it's versatile. Because you would think that being that specific, that it would be limited. I mean, how can it be that we have specific immunity that can respond to so many different pathogens? And the answer is we can respond to over 1,000,000 antigens, and actually 1,000,000 is being very, very conservative. I'm just throwing a real low number out there. We can respond to many, many antigens. We have great versatility. And thirdly, and this is the whole reason for vaccination, thirdly, is we have memory. And sometimes we call the memory responses anamnestic. And the reason is because the word amnesia means loss of memory. The prefix a or an before a word means without. So this is without the loss of memory or memory. So we have an anamnestic response. And this anamnestic response is the whole reason we vaccinate people, because it's a very violent and fast response. When I say violent, I mean violent against the pathogen, of course. And lastly, our specific defenses have tolerance. That means we recognize self and we don't attack self. So we tolerate our own antigens. But we don't tolerate foreign antigens. If we recognize it as foreign, then we fight it off. So those are the properties of our specific defenses. This is showing you that we have two main branches of our specific defenses. We have cell-mediated immunity, and the cells involved are T cells, right here, T cell. And we have humoral immunity, and this is calling it antibody-mediated, but a lot of books call this humoral, and humoral means fluid, and that's because the antibodies float around in our plasma, which is a fluid, of course. By the way, not just our plasma. The antibodies are on our mucous membranes, etc. So we have cell-mediated and humoral immunity, or cell-mediated and antibody-mediated immunity. Two branches of our adaptive immune system. This is showing the two classes of lymphocytes involved. T lymphocytes are cell-mediated immunity, and B cells are humoral immunity. Now, as it turns out, yes, B cells make antibodies, but they actually have to differentiate into a subtype of B cell called plasma cell. And then it's the plasma cell that makes antibodies, but they are a subtype of B cell. They are a differentiated B cell. Now, this is showing you another class of lymphocytes. Remember, this is innate. This is not part of adaptive immunity. That's innate immunity. Even though I'm going to talk about it again. I'm going to talk about natural killers again, because what you're going to see is that the T cytotoxics over here have a similar mechanism as the natural killers. So I will refer back to the innate immune natural killer cells.
Activation of the immune response typically begins when a pathogen enters the body. Macrophages that encounter the pathogen ingest, process, and display the antigen fragments on their cell surfaces. Macrophages with antigen fragments displayed on their surfaces are called antigen-presenting cells. An antigen-presenting macrophage interacts with a T helper cell that can recognize the same antigen. During the interaction, the macrophage releases a chemical alarm signal called interleukin-1, which stimulates the T helper cell to secrete interleukin-2. Interleukin-2 causes the proliferation of certain cytotoxic T cells and B cells. The immune response from this point follows two paths, one using cytotoxic T cells and one using B cells. Normal cells of the body that become infected can also digest some of the pathogens and display antigen fragments on their cell surfaces. The body makes millions of different types of cytotoxic T cells. Each type is able to recognize a particular antigen. The cytotoxic T cells that are capable of recognizing the antigen displayed on the surfaces of infected cells bind to the infected cells and produce chemicals that kill the infected cell. Death of the infected cells results in destruction of the pathogen. B cells also come in millions of different types, each able to recognize a particular antigen. When B cells become activated by T helper cells, they differentiate into plasma cells. These plasma cells become antibody-producing factories, flooding the bloodstream with antibodies that can bind to the antigen involved in this infection. Antibodies bind to the antigens on the surfaces of the pathogens, marking them for destruction by macrophages. Some of the B cells do not turn into antibody factories, but instead become memory B cells that may survive for several decades. Because of these memory B cells, the secondary immune response to a future infection by the same pathogen is swifter and stronger. This powerful secondary immune response is what gives immunity to some diseases after you have had them once or after you have been vaccinated.
All right, there's a couple things I want to point out from this video before we move on. So first of all, this is showing you that an antigen-presenting cell presents a foreign antigen along with a self-antigen. And that's important. So this right here is a self-antigen, and that red thing right there is the foreign antigen. Now they call this a T helper, and it is, just because they said it was. But it is in this picture too. So that T helper is going to initiate chemotaxis, that's calling other white blood cells to the area, and it's going to initiate some differentiation. And what happens is now you get a T killer. Oh, did I go too far? No, I didn't go too far. So now you get a T killer that is a T cytotoxic. By the way, yeah, it's cytotoxic T cell or T cytotoxic. Back when I was an undergraduate student, we called these T killers, but now they call them T cytotoxics. And yes, we kill the cell. So the deal is this: that could be a cancer cell, and that red foreign antigen right here is a cancer antigen, because a lot of times cancer cells express foreign antigens or abnormal antigens. So is that a good thing we're killing this cell? Well, yeah, especially if it's a cancer cell, that's a great thing. Or it could be a viral infected cell, and this red antigen that's foreign is a piece of the virus. Well, in that case, it's a good thing we kill our own cell if it's viral infected, because if we kill the cell, then the virus dies with it, and the virus doesn't have a chance to propagate and infect other cells. So there's probably a couple things that are foreign to you here. First of all, the antigen presentation. Yes, our cells present foreign antigens co-located or along with self-antigens. And yes, T cytotoxics kill these cells, but not necessarily a bad thing, because they could be cancer cells or virally infected cells. All right, now on this, what happens is our B cells have these IgD antibodies, these green antibodies sticking off of it. They're called IgD as in Delta. And there's a foreign antigen stuck to the IgD. Well, what happens is a T helper comes in and recognizes that right there. And when it does, it tells this B cell to clonally expand. That means divide hundreds of times. And not only clonally expand, but make a bunch of this antibody right here. Now, it doesn't make IgD, but what it does make has the same region out here that recognizes the foreign antigen. So it doesn't make this antibody that doesn't recognize the foreign antigen. It doesn't make this antibody that doesn't recognize the foreign antigen. It makes a lot of this kind of antibody that does recognize the foreign antigen. And there we go, all those antibodies, and they're all going to be able to neutralize those foreign antigens. Now, antibodies don't directly kill anything, but they do tag it for destruction, and that's what you see here. You see a phagocyte gobbling up the antibody-tagged antigen. And by the way, tagging it for destruction, just like we talked about with complement, is called opsonization. So antibodies can opsonize just like complement can opsonize. All right, antigen presentation. It comes in two classes. There's class 1 and class 2. First of all, and that's the self-antigens, by the way. Class 1 and class 2 are the self-antigens, because when we present foreign antigens, we always present the foreign antigen alongside our self-antigen. MHC stands for Major Histocompatibility Complex, and there's classes of MHCs. MHC class 1 is found in every nucleated cell in your body. MHC class 2 is only found on antigen-presenting cells and T helpers, for example. It's found in some lymphocytes like T helpers. So now remember, do antigen-presenting cells and T helpers have class 1? Yes, every swinging nucleated cell in your body has it. But they also have class 2, which other cells may not have. All right, this is showing you what happens when class 1 MHC molecules are used in the antigen presentation. So first of all, the big difference here is this. When class 1 MHC is used in the antigen presentation, the only cell that's going to react to this is a T cytotoxic. T cytotoxics react to class 1 MHC molecules. That's what they do. So that's what's going to attack this cell if it's presented with class 1 MHC. This is showing you how we present the foreign antigen with our self-antigen. Our self-antigen is a protein, so it's made on the rough ER, and it's packaged up. It's made in the rough ER, it's packaged up, happens to bind the foreign antigen. Then it goes to the Golgi, and it becomes a vesicle, and it floats out here and starts to exocytosize, but it doesn't completely leave the cell. It stays adhered to the membrane. So this is a class 1 MHC molecule with a foreign antigen. Now this is a class 2 MHC molecule presented with a foreign antigen. And the difference is, what's going to recognize this is a T helper. So T helpers recognize class 2s, whereas back here, T cytotoxics recognize class 1. Testable. T cytotoxics recognize class 1 MHC molecules, and T helpers recognize MHC2 antigens. All right, so who cares? I just told you what cells recognize class 1 and class 2, but what do they do with them after they recognize them? All right, let's talk about this. Well, before we talk about it, we have to talk about CD4 and CD8. On the T cytotoxics and on the T helper cells, we have these cluster of differentiation, or sometimes called cluster of designation markers. They're called CD markers. Cluster of differentiation or cluster of designation. All right. T cytotoxics have CD8s. T helpers have CD4s. You have to memorize that. T cytotoxics have CD8s and T helpers have CD4s. All right? Says it right here. CD8, T cytotoxic, CD4, T helper. What that means is CD8 responds to class 1 MHC. Remember? I said T cytotoxics respond to class 1 MHC. That means CD8 responds to class 1 MHC. And that means that CD4 responds to class 2 MHC. Remember, I said T helpers respond to class 2 MHC, and that means that CD4s respond to class 2 MHC. And you can see that right here. CD8s respond to class 1 MHCs, CD4s respond to class 2 MHCs. So that's what that means. All right, now let's get down to it. If a class 1 MHC molecule is presenting a foreign antigen, then a CD8 T cytotoxic cell can respond to it and it can kill it. This is showing you the class 1 MHC. This is showing you the CD8 T cytotoxic cell. And now what can happen is this T cytotoxic cell can release granzymes and perforins that perforate the cell and kill it. So these are going to release two things: granzymes, which are enzymes that help kill that cell, and perforins. Now, the perforins are similar to the perforins you learned in the natural killer cells. They are similar, and they do perforate, but they're not exactly the same, but they do perforate like the natural killer perforins perforate. So T cytotoxics release granzymes and perforins that kill this cell that's infected or a cancer cell. In addition, the T cytotoxics make memory cells. So the next time that you're infected with something carrying that antigen, you can attack it quicker. This is showing you the destruction of the cell. So you can see that there. And it's showing you perforin release. It's not mentioning granzyme, but there is granzymes. There is granzymes, even though it's not mentioning it. And this is the collage showing you MHC class 1 being recognized by CD8 and perforin being released and lysing the target. Okay. When MHC class 2 molecules are used to co-present the foreign antigen, I just circled the red foreign antigen, CD4 T helper cells recognize it. Now, CD4 T helper cells don't directly kill anything, but they do activate either T cytotoxics or they activate B cells. They activate both of them. So T helpers become very, very important because what they really are is a link between the humoral immune arm and the cell-mediated immune arm. So the two branches, I shouldn't say arm, you're going to think I mean your arm. No, the branches, the humoral immune branch and the cell-mediated immune branch. T helpers are the link between those two. And T helpers they'll go into a memory state. You'll have inactive memory T helpers. So they're going to act quicker next time that foreign antigen comes into your body. And they secrete cytokines, interleukins. And what these cytokines do is they activate B cells and or T cytotoxics. All right. More of the story coming up. So this is saying activation by class 1 MHC. You already know what happens. T cytotoxics are going to respond. This is saying activation by class 2 MHC. You already know what's going to happen. T helpers are going to respond. This is the collage saying what happens when class 1 MHCs are co-presenting the foreign antigen. And the answer is T cytotoxics. Don't forget the memory cells as well. So with class 1 MHC, we get T cytotoxics. We get some memory cells too. I'm not going to talk much about suppressor cells. I will tell you this just briefly. It is how we continue to recognize self as self, and we don't attack ourselves. Could something go wrong with our suppressor T cells? Yeah, that's an autoimmune disease. And of course, there's lots of autoimmune diseases, but we don't typically attack ourselves. With co-presentation of foreign antigen with a class 2 MHC, we activate T helpers.
When a virus infects a cell and synthesizes viral proteins, some of these proteins are degraded to peptide fragments. These peptide fragments are complexed with class 1 MHCs and displayed on the surface of the infected cell. Cytotoxic T cells interact with the virus-infected cells by recognizing both the viral antigen and the class 1 MHC. The cytotoxic T cell then releases cytotoxins which induce apoptosis in the infected cell and perforin which causes perforations in the cell membrane. Self-proteins are also degraded and the fragments presented on the surface via class 1 MHCs. This is not shown. Fragments of self-proteins are not recognized by cytotoxic T cells. Cytotoxic T cells remain intact, detach from the infected cell, and move on to other target cells.
Okay, then that's MHC class 1 and cytotoxic T cells.
Antigens, such as bacteria or viruses, are usually ingested and degraded into small fragments by phagocytes such as macrophages. The small fragments are then complexed with a special type of protein called a class 2 major histocompatibility complex protein, MHC, and transferred to the surface of the phagocytic cell. Helper T cells recognize an antigen when it is presented on a class 2 MHC because helper T cells have special recognition proteins on their surfaces called T cell receptors. This interaction activates the helper T cells. The activated helper T cell is now capable of stimulating B cells to become antibody-producing plasma cells.
Okay, and all of those antibodies can recognize that foreign green antigen. Whoops. That foreign green antigen right here. All of those antibodies can recognize that. All right, so what we have as a summary is MHC class 1 co-presented foreign antigens that are co-presented with MHC class 1 self-proteins are attacked by T cytotoxics. And T cytotoxics kill the cell presenting that antigen. But that's all right because it's typically a cancer cell or a virally infected cell. Whereas antigens co-presented with an MHC class 2 self-antigen, foreign antigens co-presented with an MHC class 2 self-antigen, are recognized by T helpers, which set up a chemical cascade that activates the T helper to now turn around and activate B cells to make and release antibodies that recognize the foreign antigen. And that's that section in a nutshell. See you next section.